Hello everybody, welcome to the lecture again. Today is the third lecture and we are discussing the structure of DNA and to see how chemistry plays role in determining the specific structure of DNA, which in this case is the B form of the DNA. So, earlier what we have seen is that the nucleobases, the orientation of the nucleobases. When we have the pyrimidine nucleobases, thymine and cytosine, and the purine nucleobases, adenine and guanine, which conformation they exist, which form they exist. And we have seen from the, I have given the full chart of the pKa values of the individual nucleobases for their individual heteroatom positions, nitrogen positions. And from here you can understand that for all these nucleobases, the keto form which is given in this fashion, keto form, keto form, keto form and the amine form, keto and the amine form. Here also is the keto form and the amine form, here is the amine form. These are the actual forms that will exist under the physiological condition, that is at pH 7.4, and that you can explain from their experimental pKa values. If you take the enolate forms or their imine forms for the amine groups, you will see that those forms are not stable at the physiological pH at pH 7.4, and the keto form and the amine forms are the stable form. So, in DNA the nucleobases will exist in this form, the structure of the nucleobases will be this. So, which is a kind of log structure, and that is of course responsible for the Watson-Crick base pairings, for these AT base pairs with 2 hydrogen bonds and GC base pairs with 3 hydrogen bonds. If you look at, if you take the enolate forms, the hydrogen bonding pattern will be entirely different, and that does not happen usually. And similarly, we have seen the other type of rotation, the locked rotation of the nucleobase when it is attached with the sugar, deoxyribose sugar. So, this is the syn form, where I have explained that with the double bond oxygen here, it is closer to the oxygen of the sugar. And similarly, for the purine bases, for adenine, the 6-membered ring is just right on top of the sugar moiety, and both of these forms are called the syn forms, and they are not stable because they have huge steric crowding and the electron repulsion factor. So, therefore, the stabler form is they will flip, they will rotate around the CN single bond, because the rotation around a single bond is favorable at room temperature. So, it will rotate and give you the anti-form, which is the better form or the more stable conformation for the DNA. So, again, so this conformation and this conformation here will be the favorable conformations that we see. Also this conformation is present in DNA, and that is the explanation why. Of course, this has also that plays a vital role in forming the base pairs. So, if you see that 6-membered ring here is out, is a little bit away from the sugar moiety, R is the sugar, and that is why it is available for hydrogen bonding. So, next comes, today I will talk about the other 2 factors that are responsible for the helical structures of the B form of the DNA. One is the phosphate, the orientation of the phosphate, and how the skeleton, basic skeleton of the DNA looks like, or which orientation it will assume. So, if you look at this structure, here is the sugar and the base, and here is the 5' end, and this is the 3' end of it. So, here if you see, it starts from the phosphate P, this is O, one is double bond O, one is O minus. So, O, P, O, and this is the 5' end of this sugar, and then there is a 3' end of the sugar with a oxygen that forms a bond with the next phosphate, and it goes on like this. So, there are 2 planes in this case, one is this, other one is this. So, that is called, so 2 planes, and when there are 2 planes there is angle between the 2 planes, and in this case the favorable orientation will have a dihedral angle, DAA means dihedral angle. Favorable dihedral angle would be minus 60 degree. That comes from the Gauss effect, as can be seen here. So, if you see, if you consider this bond P and O, so P and O has a single bond with an electron cloud here, and then here in the next one you have the oxygen, free oxygen with a lone pair of electrons. This is the favorable orientation for this case, with the lone pair of electrons away from the sigma bond, because otherwise if it is closer, then there will be a bond pair lone pair repulsion, and that is not favorable. So, this is the favorable structure. Similarly, if you look at this bond, take this bond as a sigma bond, and then lone pair of electrons on this oxygen, they will be away from the sigma bond to avoid bond pair lone pair repulsion. And that is how the structure or the angles in the backbone or the phosphate skeleton comes from in DNA. Another aspect is that when you see PO bond, this PO bond or this PO bond, of course they are single bonds, but these are not entirely single bonds. They can undergo resonance. If you consider this a double bond, then there will be resonating structures possible for the phosphate, and that makes partial double bond character of this PO, as well as the same thing will happen in this case also, resonating structures. So, although these are single bonds, free rotation around the single bonds should have been really possible and should have happened, but there are somewhat restrictions is there. And the overall the Gauss effect as well as the resonating structures, they actually lock the conformation the local conformation of the skeleton in this fashion. And now coming to another important aspect, which is the conformation of the sugar. What orientation the sugar moiety in DNA or in RNA will have. So, to start with that, now just consider without the whole DNA structure, consider the deoxyribose sugar. This is a pure deoxyribose sugar, deoxyribose sugar, which is present in DNA with the OH below the down the plane and the 5' OH up the plane. If you consider RNA, RNA is D-ribose sugar, where at 2' position you have another hydroxyl group down the plane. So, if you do the numbering, it is like this: 1', 2', 3', 4', 5', 1', 2', 3', 4' and 5'. Now, if I go back to the DNA structure, the paper mode, you have seen the sugar is oriented in these directions. So, now if you look here in this part, it is not actually on the plane of it. I have a rough draw of the 5-membered ring or 5-membered sugar with the oxygen here. So, if you see, the sugar actually exists in these fashions with the oxygen folded up the plane. If you consider this is the plane of the sugar, then the oxygen would be up the plane. So, it looks like an envelope which you can fold this. So, this is the closing part of the envelope and this is the plane. So, that is what the 3D structure of this sugar part is. It will be out of the plane and it will be in this direction. So, here also the same thing, it has a envelope form, the ribose sugar moieties. And now if you consider the DNA, this is oxygen. So, this is 1', this is 2', 3', 4'. And if you look at the deoxyribose, so there is nothing here, there is just 2 hydrogens. In the 3' positions you have one hydroxyl group and that is down the plane. At 4' position you have the CH2OH that is up the plane. Okay. 1' position I am not defining it because the nucleobase comes, and nucleobase comes usually from the up the plane actually. Here it should be up the plane, here also this should be up the plane. When the nucleobase comes, it is up the plane. So, now coming to the orientation. So, if this is the structure, then what kind of orientation it can have. So, since this is a sugar and 5-membered ring, it can have the puckered conformation. So, here I have written the 2 sugar pucker conformations. I will come in details there. So, if you consider the deoxyribose and if you draw the sugar pucker conformations with this, then it will look like this. So, when you draw this as a pucker conformations, you can twist the around the the 2' and 3' position, you can keep the plane 1' oxygen, 4' 5' ready in one segment, and then twist the 2' and 3' bond, and that twisting will give you different conformations. So, one is that if I twist it at the 2' end, it will look like this. So, this would be up the plane, this is in front of you. This is in front of you and this is on the plane. And of course, now I am considering with the nucleobases. Nucleobase is up the plane. So, up the plane here in this case I am writing base for nucleobase. Up the plane in this case is equatorial position, because the axial position is H. This is the 2', this is 1'. 2' position, there is no hydroxyl, both are H, axial is H, equatorial is H. 3' position, you have one hydroxyl group here, and that is down the plane. So, down the plane has to be this is down the plane, which is axial, equatorial is H. Now, CH2OH is of course up the plane. So, up the plane is kind of axial, equatorial is H. So, this would be the conformation, the Parker conformation of the deoxyribose if you consider if you twist it at the 2' end, and this is known as 2' endo twist. The same thing, if you twist it back, if you twist it at the 3' end, then it will look like this. This is bold because it is in front of you, it is towards you, and here is the base, and this is the 3'. So, you have twisted it in the 3' position. So, now the base is up the plane, and up the plane in this case is axial. Here 2' nothing, 3', 3' you have OH down the plane. So, this is down the plane, which is equatorial is down the plane, axial is H, and this is the 5' up the plane is your hydroxyl CH2OH, down the plane is H. So, this is called C3' endo twist. Now, out of these two structures, which one is more favorable, that we have to find out. So, if you look here, the base is in equatorial position. Here you have the hydroxyl group in the axial position. Axial position substitutions are usually not favorable, and here also axial position, but here the hydroxyl group is little bit away from the carbon. So, that does not have much interactions, much steric interactions with the sugar. This hydroxyl, however, will have 1,3 Gauss interactions. So, out of this, which one is more favorable? So, if you look at the DNA, then, and there is another factor that I will talk about. So, C2' endo twist, which is this form, will be the predominating or the favorable form for DNA, because although you have this is axial position, the nucleobase present here is better off, because it does not have, in this case if you consider the axial base that has lot of lone pair repulsion and the steady crowding within the other group, within this and within this region also. So, that is the disfavorable part of it, and in this case nucleobase is in the equatorial position, which suffers less interactions or less crowding and less repulsion. So, another thing is that if you look at the phosphate, here will be a phosphate, here will be a phosphate, and then the distance between the two phosphate groups in this case is around 7 angstrom. That is, if you have a phosphate instead of H, if you have P, instead of H, if you have P. then this distance is 7 angstrom. On the other hand, this distance, if you have P here and if you have P there, this distance is around 6 angstrom, close to 6 angstrom. So, this is closer, and closer means they will have to face more steric interactions or repulsions. So, for the DNA conformation, the C2 prime endo twist is the favored conformation. Now, if you look at the RNA, that is the ribose sugar. Ribose sugar was this. Here comes the base up the plane, and here you have the hydroxyl OH down the plane, OH down the plane, up the plane, and then CH2. This is up the plane. Now, the same thing if you draw the C2 prime and C3 prime endo twist. This is the oxygen. This is towards you. So, base is here, equatorial, and here you have the 2 prime position, you have a hydroxyl group, and that is down the plane. So, down the plane has to be equatorial, rest are all the same, and this is kind of axial. So, that is your C2 prime endo, base is up, here which is down the plane, so axial, this is down the plane, equatorial, and this is up the plane, equatorial. So, that is your C3 prime endo twist. Now, if you compare for the ribose sugar RNA, then you will find that here you have a strong repulsion between the base and the hydroxyl group here. And in this case, although the base is in axial position, that actually reduces or that decreases the huge repulsion that comes in this case between the hydroxyl group and the base. So, in this case for the RNA, C3 prime endo twist will be the favorable structure, and that overall changes the conformation of the total RNA. So, DNA, as I have said, has the B form with proper double helical structures, and that is coming from here, because if you have seen C2 prime endo twist for DNA, you can have this is the 3 prime end, you can have the extension in this way, and the 5 prime end will go that way. So, you will have a proper skeleton that is known as the B form. In A form, your extension has to be in this part, and from the 5 prime end this part, and that will give you the A form of RNA. So, RNA mostly has A conformation and DNA has B conformation, and that is one of the reason, of course, it is coming from because of the different sugar Parker conformation of the DNA and the RNA. So, these are the factors that play important roles in determining the specific structure of the DNA, as I have talked about again and again. That is the B form of the DNA with the double helix structures, and that is responsible for the proper hydrogen bonding, which we usually write in these fashions: AT base pairs and the GC base pairs. So, this is the most double helical structures. Apart from these, so AT base pairs and GC base pairs are usually called Watson-Crick base pairs. So, apart from those, there are some non-Watson-Crick base pairs also. They are sometimes naturally available. At the same time, when we make new molecules, when we make modifications of the nucleic acids, then the non-Watson-Crick base pairs play a vital role in making the new kind of substitutions or new kind of molecules. So, naturally also apart from Watson Crick, there are other types of base pairs that are found. One is called the Hoogsteen. So, if you look back at the AT base pairs, you see that for adenine, I have missed the nitrogen again here. AT base pairs, you see the 6-membered ring of the thymine. So, this carbon and this NH proton are involved in hydrogen bonding. On the other hand, if you look at the adenine side, it is all the 6-membered ring that is involved in the hydrogen bonding. NH, this NH and this NH2, both are coming from the 6-membered ring. The 5-membered ring is not involved in hydrogen bonding. Similarly, if you see in the GC base pairs, of course, cytosine has only one ring and all 3 of them are involved in the hydrogen bonding. But if you look at the guanine, it is only the 6-membered ring. The substitutions on the 6-membered ring are involved in the hydrogen bonding, not the 5-membered. Now, if you look at the Hoogsteen hydrogen bonding, this is R is sugar again. Both the 5-membered and the 6-membered are involved in hydrogen bonding, and as usual this is for the thymine part. So, here this will totally change the orientation of the whole DNA, because it has to I mean its minor groups, major groups, minor groups, everything will be changed. Also the structure of the DNA will be changed. So, this is a unnatural, it is also found sometimes in nature as well. This is the non-Watson Crick base pairs, one kind. Other is the reverse Hoogsteen. Reverse Hoogsteen here is, if you turn the 6-membered ring thymine a little bit, make the sugar here, then basically it's this and this are present, are involved in hydrogen bonding with the Hoogsteen mode, taking the 5-membered ring as well as the 6-membered ring. So, this is called the reverse Hoogsteen. It is basically, if you think, is this and this atom are this. So, these two are involved in the hydrogen bonding. Both are 2 hydrogen bonds, AT base pairs. GC Hoogsteen and GC plus Hoogsteen. Cytosine sometimes exists as C plus, and then it can undergo hydrogen bonding in a Hoogsteen mode. Hoogsteen mode means again the 5-membered ring is involved. So, this is called the GC plus Hoogsteen. And the details of this kind of hydrogen bonding are actually written here, so you can go through it. And there is third type, which is called the oval base pairs. Oval base pairs are kind of sliding, whatever the normal base pairs you have seen, AT, if you just slide them a little bit, this one, if you slide it a little bit, then what the orientation you get and the hydrogen bonding you get is known as the oval base pairs. So, in this case, the thymine has been slided down a little bit, so that you have now this amine is not coming into the base pairs, you have only 2. So, this is guanine and this is for uracil. So, GU base pair, that is actually present in nature in tRNA, this kind of oval base pairs are found. Now, Hoogsteen base pairs are mostly they mostly occur in DNA triple helix. Now, usually DNA we know they are the double helix structures, but sometimes DNA can undergo triple helix formations with 3 strands, that we will see later also, that when we do the modifications we can see how the triple helices are formed. So, if you have 5' to 3' DNA and then 3' to 5', then they take one of these one strand as the key strand and forms another base pairs. So, here you have one set of base pairs, and with this you have the other base pairs with a third strand, that is 5' to 3' end. So, this is Watson Crick base pairs, 6-membered ring versus 6-membered ring, and this part, the other part is the Hoogsteen base pairs, where the 5-membered and the 6-membered both are involved. This is Hoogsteen. So, that is how the DNA triple helices are formed. So, we have seen how the structure of DNA looks like. Now usually when you write a DNA, then as I have mentioned before, we write it in 5' to 3' orientation. 5' is phosphate and 3' is hydroxyl. When you write a DNA double strand, the convention is you write it in this fashion, 3' and 5', and then you write the sequence of the DNA. I am just writing an arbitrary sequence, A T G C, A T G C, A T, and then of course the sequence of the other strand will be the complementary of it. So T A C G, T A C G, T A. That is how our DNA double strand is usually been written. Now in paper when you write, then you do not have to write the whole double helix sequence, double strand sequence. If you write only one sequence, 5' to 3' strand, then it is understandable what will be the other strand. So you can write either of this sequence. So that is fine. That is what is being usually used to represent the DNA. Now depending upon the sequence of the DNA, DNA has different strengths. For example, if I just take a DNA which is all AT, A, 5', 3', T, 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 3', 5', and so we are talking about two properties of DNA, that is called DNA melting and DNA hybridization, 5', 3'. Now if you consider all GC, obviously they will have different strength, because AT base pairs have 2 hydrogen bonds, GC base pairs have 3 hydrogen bonds. So GC is stronger, this double strand would be much stronger compared to this double strand. So there is a way to determine the strength of the DNA, and that is known as DNA melting temperature. DNA melting temperature. In short, this is written as Tm. Now this is like any other melting point that you know. If you have a compound, melting point means the 50% of the compound should melt away. So for DNA case, it is the melting temperature is the temperature at which the 50% of the double strand is melted away into the individual single strand. So here will be all A, here all T. That is called the denaturation. So is that melting temperature is the temperature. So if I define Tm, Tm is the temperature at which 50% of the double stranded DNA, DS means double stranded DNA will be denatured. Denatured means melting away, the breaking of the double strand, because it is against the nature. So doubles will be denatured, temperature at which 50% of the double stranded DNA would be denatured. So that is the definition of the melting point, and it's kind of a characteristics of a DNA, not really, but it gives you an idea how strong is the DNA or what can be the length also of the DNA. So obviously now if you look at the AT rich sequence and GC rich sequence, the melting temperature of the GC rich sequence will be higher, because you need more heat or more energy to break the GC sequence, GC rich hydrogen bonds. So 50% here, and here you will have 5 prime to 3 prime all G plus all C. So this is important actually, because when you synthesize, sometimes we can nowadays manufacture short sequences of DNA, or if you isolate a DNA sample from somewhere else, you need to know what is the strength of the DNA sometimes if you break them into pieces. Now the second question is how to determine, how to know what will be the melting temperature. How do you know at which temperature the DNA is getting denatured? The point is that DNA, like you measure the melting temperature of the compounds, because they have visible quantity, you can see them, and you can take them in a capillary, measure it, and you can see them getting melted away. That is not possible for DNA, because the sample of DNA, when you isolate a sample of DNA from somewhere else, the concentration is so low that sometimes you do not even see them. They are less than milligram quantity, sometimes to the level of microgram also. So with those small quantity, you cannot obviously weigh them, and you cannot see them transforming from the double stranded DNA to the single stranded DNA. So how do you determine the melting temperature of a DNA? We usually do it by UV spectroscopy. So the DNA contains the nucleobases adenine, thymine, guanine, cytosines, and as I have mentioned, all of them are aromatic and all of them are heterocycles. So they absorb hugely in the UV visible region, ultraviolet region. So if you do a absorption, UV absorption study, UV visible absorption, UV visible absorption, because this is one of the technique which is very sensitive, which requires very small amount of sample to give you a very good signal. So that measures, it's a spectroscopic technique where you can measure how much the light is absorbed by the sample at which wavelength. So then you will see that if this is your absorbance and this is the lambda value, the wavelength, then you will see the nucleobases, they absorb at certain wavelength. For example, I will just give you one example. This one is, so it's kind of 260. So this maximum lambda max, this value is 261 nanometer. So lambda is in nanometer. At 261 nanometer, you have maximum absorption if you have adenine. So I'll give you a chart where the individual nucleobases absorb. So this is base for nucleobase, and this is your lambda max, where you have the maximum absorption in terms of nanometer. If you have adenine, it absorbs maximum 261 nanometer. If you have thymine, that has a maximum of 264 nanometer. If you have cytosine, it's around 267 nanometer, and guanine has 276 nanometer. So they are more or less close, and on average we consider that if you take the absorption of DNA, you will see a sharp peak or lambda max at around 260 nanometer. So 260 nanometer is kind of a characteristic peak of DNA, and that has the absorption maximum for the whole DNA. That is taking average of all the sequences. So if you can measure the concentration of a DNA by measuring the absorption value, and therefore if you know, now if you go on, come back here, when you have the double stranded DNA, the two strands are intact and it will show you the certain absorption value at 260 nanometer. Now, if you increase the temperature, this is in solution water or buffer. If you increase the temperature and reaches the melting temperature where the two strands will be separated away, then what happens is you break all the hydrogen bonding interactions and you break also all the pi stacking interactions. So, all interactions are broken, and as a result the absorption of these individual nucleobases gets increased, because when you had the double strand, when there was the hydrogen bonds, then the electrons were moving towards the hydrogen bond also, or they were involved in the hydrogen bondings, and that decreases the absorption or absorbance. Now, once the nucleobases here gets free, becomes free, then they can behave as the original single molecule entity and their absorbance gets higher. So, you will see a increase in absorption on increasing the temperature, and there will be this kind of curve that you have slowly increasing the absorption, then there will be a sharp rise. So, here is the temperature and here is the absorbance at 260 nanometer. So, at the beginning when the temperature is low, because the hydrogen bonding were still intact, so the amount of increase was low, and when you break all the hydrogen bonds, when it becomes free, two single strands, then suddenly the absorption gets increased, jumps up and then becomes saturated, because now they are all single molecules. So, half 50% of it, the half point here, this will be your melting temperature. So, that is how you determine the melting temperature of a DNA. Another aspect I should talk about in the DNA is the solubility, that I forgot to mention. So, when you look at the DNA structures, of course, this is phosphate, means there is double bond O and it's O minus. So, DNA overall is negatively charged because you have the phosphates, inorganic compound, the phosphates, and in ionic form, O minus form. So, it is basically the ionized version of phosphoric acid. Now, if you look at the nuclear base, this is neutral, sugar is neutral, only phosphate is ionized and it is negatively charged. So, overall DNA is negatively charged, and that also make it soluble in water. This is the positions where water molecules bind, or when you have salt molecules will stabilize the counter anion. So, you have one acidic component, this is the acidic component, and nucleobase is the basic component. That makes DNA soluble in water pH 7.4, or either water pH 7 or in buffer physiological condition, that is pH 7.4. Now, coming with DNA hybridization. DNA hybridization, or you can call it annealing. So, we have seen how DNA gets denatured from double stranded DNA to single stranded DNA. Now, the reverse one, if you have two single stranded DNA, how do you make a double stranded DNA? That is called the DNA hybridization. So, if you have two single stranded DNA in water or in buffer solution at pH 7.4, let us say, you mix them together, and then they always do not form the perfect double helix that you would anticipate. So, you have to do certain changes to the solution, and that will give you the proper hybridization of a DNA. So, that the reason is, if you have one single stranded DNA plus the other single stranded DNA, so at room temperature, if you just mix them together without doing anything, then what happens is there are already within the same DNA strand, there are small interactions, certain interactions are present. For example, it can have the self hybridization, partial self hybridization. Some part of the DNA may be complementary to the other part, and they can hybridize, making a folding structure of DNA. Not much stable, but still stable at room temperature, sufficient. The melting temperature is higher than room temperature. So, at room temperature they will have the self hybridizations. Similarly, for this strand. Second is that, of course, DNA, as I mentioned, is negatively charged, phosphate. So, if you want to bring the two strands together, they will simply repel. So, the hybridization strength or the strength of the double strand, if you just bring two single strands together, will not be high, because the phosphate negative charge repulsions will be there, as well as the self hybridizations will be there. So, in order to avoid that, what you have to do is, if when you take the two single strands, usually you have to use a salt. Either we use sodium chloride or magnesium chloride to stabilize the counter anions. Now, once you use the salt, then they are stabilized, and if you mix them together, the two single strands, then they can form perfect hybridizations. And second, you have to heat it up at higher temperature. We usually do at 95 degrees Celsius temperature. That is the usual practice to break all the shorter interactions or all kinds of interactions that were present within the same molecule. So, once you heat it up at 95 degrees Celsius, then all interactions are broken. Now, you mix them together and allow it to cool down at room temperature. To room temperature. Or if your DNA is shorter strength, you can still move to lower temperature, just like refrigeration, 4 degrees Celsius. Now, if you do that, then your thermodynamics is playing role. This is cooling down is a slow process. So, that will now your thermodynamics will dictate the strength of the DNA. So, whichever the best strength, in this case of course the double strand, that will have the best strength, will be the most favorable process. So, therefore, all the shorter hybridizations or partial hybridizations like this, so partial hybridizations will be thermodynamically unfavorable. The full hybridization, if you have the complementary strands, will be thermodynamically favorable process. So, this is some aspects of DNA regarding the synthesis of nucleobases and the synthesis of the whole DNA, that we will discuss in the next module. So, now so much so for the DNA, and I will start a brief introduction on the amino acids, peptides and proteins. So, details of the synthesis of the nucleobases, sugars and the phosphate backbones we will talk later in the next module. So, amino acids are part of protein. Protein is constituted of amino acids, or the peptides are constituted from the amino acids. Amino acids has a general structure as they have a carboxylic acid group, they have one amine group, and they have a R group, which is called the side chain, and that actually varies mostly from amino acid to amino acid, and here there is a H. So, this is the primary constitutions of amino acids that make up the structure of the proteins or peptides. So, when you have two amino acids, I am just writing flat R NH2 R1 NH2 COOH, and you fuse them together, condense basically, remove a water molecule, then you get a different bond, an amide bond, this is the new bond that has been formed. That is basically an amide bond, but since this is present in the amino acid environment, we call it, we have given a specific name, we call it a peptide bond, and that is how came the name peptide. So, when you increase this process further, you have a chain of the peptide or chain of the protein composed of peptide bonds. And just like we have seen for DNA, we have named it from 5' end to the 3' end. the peptides or the proteins are usually terminated as N terminal and C terminal. So, if you increase the length, you will see the left hand side will always have amine free. This is called the N terminal. The right hand side you have a carboxylic acid free. This is known as C terminal. The stereochemistry of the amino acid is in this fashion, which is usually has the L configuration, or this is equivalent to S configuration if you see the priority sequence. So, left handed and S configurations. This comes obviously from the D and L comes obviously from the glyceraldehyde, where you have a CHO, H, CH2OH and OH. OH is in the left, the heteroatom is in the left hand side, that has the L conformations. This is called L glyceraldehyde. If you draw this into the Fischer projections, you will see that this has a structure, this has the projection of, this is the length, the longest chain, amine in the left hand side. So, this is called L. So, amino acids are of L configurations, and L means S in this case. So, R varies. As I have said, there are around 21 naturally occurring amino acids. It is really 20 abundant in nature, and one is seleноcysteine, which has this structure, which is sometimes available in cells, but it does not code for a protein. Thank you.